Everyone, my topic is ultracentrifugation, and the basic principle behind centrifugation is that we use centrifugal forces to separate and purify the mixtures of biological particles in a given liquid medium. It is a key technique for isolation and analyzing the subcellular fractions or different components of the cellular mixtures, for example, proteins, nucleic acids, etc. The analytical centrifugation is mainly concerned with the study of the macromolecules or isolated supramacromolecules or supramacromolecular assemblies of the given mixture. Preparative assemblies, Preparative centrifugation methodologies is devoted to the actual separation of the tissues, cells, subcellular structure, membrane, vesicles, and other particles of a biochemical interest. Types of centrifuges. Many different types of centrifuges are commercially available. That includes large capacity low speed preparative centrifuge, refrigerated high speed preparative centrifuge, analytical ultracentrifuge, preparative ultracentrifuge, large scale clinical centrifuge, and small scale microfuges. The topic of our concern are the analytical centrifuge and preparative ultracentrifugations. Ultracentrifugation is a centrifuge optimized by spinning the rotor at a very high speed and capable of generating acceleration as high as 100,000 Gs, approximately 9800 km per second square. There are two types of ultracentrifuges, that is preparative and the analytical. The both classes of instrument find very great significant use in the molecular biology, biochemistry, and polymer sciences. In 1925-26, Berg constructed a new ultracentrifuge that permitted fields up to 100,000 Gs, that is 42,000 rpm. The vacuum ultracentrifuge was invented by Edward Girdon in the physics department of the University of Virginia, which further reduced the friction of the air and enhanced the speed and the round per minute. In 1946, the Pickle co-founded Spinco to market of analytical and preparative centrifuge based on his designs. The ultracentrifuges have different types of rotors. The ultracentrifuges are available in a wide variety of rotors suitable for the great range of experiments. Most rotors are designed to hold test tubes that contain samples. Swinging bucket rotor is the first one, in which the tubes are hung or hang so that the tubes reorient to the horizontal axis when the rotor initially accelerates. The fixed angle rotor, it does not change its angle. It has a fixed angle. It does not change its orientation when the rotor starts to accelerate. The zonal rotor are designed to contain a large volume of sample in a single central cavity rather than in different tubes. The vertical rotor is fixed cavity and a straight fixed alignment to the centrifuge rotor's axis. This picture demonstrates three types of rotor. The first one is the fixed angle one, second is a straight or the no angle centrifuge, and the other last one is the swing bucket rotor. The first one of topic is the preparative ultracentrifugation. The cellular and subcellular fractionation techniques are indispensable methods used in biochemical research, although the proper separations of many subcellular structures is absolutely dependent on the preparative centrifugation, and the isolation of large cellular structures, nuclear fraction, mitochondria, chloroplast, or large protein precipitates can be achieved by conventional high-speed refrigerated centrifugation. There are two types of preparative centrifugation. First, differential ultracentrifugation, and another one is the density gradient ultracentrifugation. The differential centrifugation is based on the difference in the sedimentation rate of the biological particles of different size and density. Crude tissue homogenate containing organelles, membrane, vesicles, and other structural fragments are divided into different fractions by stepwise increase of the applied centrifugal fields. The biological structures or aggregates are separated into pellets and supernatant fraction depending upon the speed and time of the individual centrifugation steps and the densities and relative size of the particles. In the case of the rigid biomolecules and the structures such as muscular and connective tissues, the debris pellets is rehomogenized and centrifuged. This is done multiple times to remove the impurities and the different mixtures. This is also done in the samples of human biopsy. Initially, all particles of homogenate are evenly distributed throughout the centrifuge tube and then move down the tube at the irrespective sedimentation rate during the centrifugation. The largest class of particles form the pallet at the bottom of the centrifuge tube, leaving the small size structure within the supernatant. Some smaller particles or the constituent of the mixtures are tapped in the pallet. Therefore, earlier stated point of rehomogenization and ultracentrifugation proves to beneficial. The density gradient ultracentrifugation is used for the isolation and purification of the ultracellular vesicles for cell culture media and biological fluids. Density gradient separation involves the centrifugation of biological samples through a centrifugation medium of higher or greater density. When exposed to an elevated centrifugal forces, the sample component migrate through the medium and separate based on their buoyant density. This results in the purification of EB population through the separation from the contaminating protein and protein RNA complexes. The cesium chloride is widely used for bonding of DNA and isolation of plasmids, nucleoproteins, and viruses. Sodium bromide and sodium iodide are employed for the fractionation of lipoproteins and banding of the DNA and RNA molecules respectively. Various companies offer a range of gradient molecules for the separation of whole cells, subcellular particles, example percol, fecol, dextran, metrizamide, and nicotins. The last other topic, other part of the presentation is analytical ultracentrifugation. Analytical ultracentrifugation refers to the analysis of the micromolecular solution by its subjection to the gravitational forces up to 3000-fold greater than the gravity. Analytical ultracentrifugation has been employed to characterize the mass, size, shape, and association properties of the macromolecules in the solution. The techniques has been broadly applied to research problems in biochemistry, molecular biology, and polymer sciences have also found practical application in the pharmaceutical and biotechnological industries. This is a general demonstration or graph demonstrating the principle, structure, and functioning of an analytical ultracentrifugation, in which X emission has been generated from a source and it's been passed through the medium and analyzed by the photomultiplier tube. Some of the most attractive features of analytical ultracentrifuges are versatility. A wide variety of samples can be examined by AUC, including molecules ranging in size from sucrose to virus particles. Rigor. AUC experiments are directly interpreted in the context of the thermodynamics and hydrodynamics theory, so it is not necessary to run standards to calibrate each experiment. Also, because of the experiments are performed in a free solution, there are no complications due to interaction with the matrices or surfaces that can complicate interpretation of other types of measurements. Convenience. Recently, a new instrumentation and data analysis methods have been made AUC much more convenient and accessible to the general biochemistry and polymer science communities. In the contrast to the earlier experiments, and the instruments are easy to set up, and centrifugation parameters and data acquisition all under the computer control. In addition, the power desktop computers are now software have greatly accelerated the data analysis process and produces a great significant results. The sedimentation equilibrium variant of the analytical centrifuge retained supremacy for many years as the benchmark standard of the molecular mass measurement for the homogeneous proteins. A role now have been taken over by the mass spectroscopy. The comparative function of analytical and preparative centrifugation. The function of the analytical centrifugations are uses a small sample size, build an optical system to analyze progress of the molecules during centrifugation, uses relatively pure sample, used to precisely determine sedimentation coefficient and MW of the molecular weight of the molecules. Bachmann model E is an example of centrifuge used for these purposes. Functions of preparative centrifugation. Large sample size can be used. No optical readout collect fraction and analyze them after the run. Large pure samples can be used to estimate sedimentation coefficient and molecular weight. generally used to separate organelles and molecules most